Everybody, it's Mr. Dan. In this video here, I'm going to take you through lesson 2A, which is going to be all about transcription and translation. Okay. Before we get into that, let's quickly look at our overall learning goal, just so we can make sure we're in line with what we're supposed to talk about. So we want to construct an explanation on evidence for how the structure of DNA determines the structure of proteins, which carry out the essential functions of life throughout systems of specialized cells. So we're talking about how structure of protein turns into structure of how structure of DNA turns into structure of proteins, which turns into function of cells. All right. So starting off here, just like this is a look of something we'll see a little bit later, but this is DNA and the beginning of a very important process that we're going to talk about. Oops. Let's go this way here. So we're talking about a thing that we mentioned in class called the central dogma. Okay, and so what this is it's essentially just moving from DNA to RNA to protein through processes of transcription and translation. We'll get into replication a little bit in a later lesson, but for now, DNA to RNA to protein, transcription and translation. So how do we do that? This is a video that you can watch on your own. I'm not going to play it right now, but it's a great animation to show you like a real time the processes that are going on. So please do take the time to watch this video right here. I'll make sure to share the presentation with you. So let's get started here. We want to talk about transcription and translation today, two super important processes. So this is a picture right here of a cell. This picture is in my classroom. You can come look at it anytime. It's an E. coli cell. E. coli they're a type of bacteria. You can find them in soil, in your digestive tract. They're super common. There are some that cause disease and some that make you sick, and some that aren't so bad. But anyways, this is a bacteria cell. Different than plant and animal cells, but it's got what we're talking about for this lesson, so it's going to work just fine for our purposes here. So transcription and translation. This is something here, like it all starts with DNA. Okay, so maybe you can see where the DNA is in this cell. I hope so. It's down here, the yellow ropes and stuff. That's DNA. One of the things that makes a bacteria cell different than a plant or an animal cell is that they don't have a nucleus. They still have DNA, but it's not packaged in a membrane like ours is. Okay, so it starts with DNA, and then that DNA, it needs to be opened up before it can be read. DNA has that double helix twisted ladder form, and so you can't access it until you open it. And so we're going to focus on this area right here. Once it's opened up, RNA Polymerase, which is a special enzyme, it's going to come in and it's going to make a copy of that gene. And then that copy called mRNA is going to be, it's going to make its way to a ribosome, which are these purple things all in the cell space here. And once it connects with that, then the ribosome can translate that mRNA into a protein. So this is the basics of it, and we're going to look closer at the individual steps along the way here. Okay, so starting off with the DNA, that was our whole last lesson. What is DNA? So here's a slide that gives you a bunch of information. The sugar phosphate backbone with the bases coming off of the sugars. Here's the different types of nitrogenous bases that DNA has. Talking about the how everything's held together. So you guys can come and look at this, even a bit of how like DNA and RNA, how they compare down here. So this is a great little read for you. I encourage you to check it out. So we know DNA, and now let's get into the real juice of it here. Step number 2. Let's talk about this molecule here, which is called RNA Polymerase 2. There's a polymerase 1 and a polymerase 3, but number 2 is the one that we're most interested in here. Okay? So the job of this protein is to open up the DNA. As you can see here in this picture, the green double strand, that's the DNA. So the job of this polymerase is to open it up and to begin making a copy of just one side of that DNA. Okay? It's going to find a particular gene that it's looking for, and it's going to open that gene up and start copying it. Okay? That gene codes for a specific protein. So the polymerase here, take a look at this picture, and you see how it's got a couple different colors. It's blue, it's light green, it's dark green. And the reason for that is because this is actually a complex or a combination of a couple of proteins, and they all come together to make this greater thing called polymerase. It needs to be complex because there's different parts of it that do different things. This whole polymerase, it's gonna slide down the DNA. It's gonna open up the DNA. It's gonna begin making copies of the DNA. So it's got a lot of different jobs to do, and so it needs different parts in order to do that. In the end, the result of this is going to be a piece of mRNA, which is this red piece coming through here. It's a single-stranded piece of nucleic acid. It's much shorter than DNA. But that's going to be the product that we're going to get out of it. So this is RNA polymerase, a really important enzyme for you to know about. Please write down that name. Please know what RNA polymerase is. So that's what's going on here. That's what we see, this white piece coming out. That's the mRNA coming off of the DNA here. So what happens next? Well, let's look a little bit closer at the actual process of transcription. Okay, so here's a simplified version of that polymerase, this purple oval here, and here's our DNA, and we again see the job of it unwinding and then creating the RNA here, specifically mRNA, which stands for messenger RNA. So a messenger, right, it takes information from one place and it delivers it to someone else, and then that someone else does something with that information. So mRNA is what we're making. So the important thing that it's doing, right, in creating this message, it's collecting nucleotides from within the nucleus or within just like the middle of the cell if it's a bacteria. And these things, these nucleotides, they're floating around all over the place, right? In the nucleus, that's where DNA is. DNA is made out of nucleotides. So there's a lot of extra parts laying around. And so it's finding these. They're everywhere. It only needs one of four, so it's going to find the right one. It's going to attach it to one that matches with the code here, right? So we're matching A's and T's and C's and G's, or rather, I should say, A's are matching with U's, T's are matching with A's, G's are matching with C's, and C's are matching with G's. But when we make this mRNA, remember they use uracil instead of thymine, so it's slightly different. But here's what's going on. We're making a copy of the DNA, just a piece of it to create one copy of a gene. Okay, so that's transcription for us. And that's what's going on here. So that's how this thing is getting made within the polymerase enzyme. So then the next stage is that we're going to go trace that. We're going to trace that mRNA up to a ribosome. And so in a prokaryotic cell or in a bacteria here, there is no nucleus, but that mRNA, it does need to leave the center area. It's very dense with DNA, so there's not much other than DNA going on in here, so it needs to leave that area. If it were to be a eukaryotic cell or a plant or animal cell, then it would have to leave the nucleus area and enter into the rest of the cell. So here we go, number 4. First thing we're going to look at is just ribosomes in general, and I'm going to give you a couple different views here. Like the polymerase enzyme, a ribosome is a complex or a combination of a bunch of different proteins, and so this is multiple views of one ribosome here, side view, bottom view, sort of an angle here. But you should notice a few different things. There's multiple different colors. The different colors indicate different pieces or different chains of proteins, and they're all fused together. We have a big top section up here and then a smaller bottom section. So this has two subunits to it. You can see there's a hole right here, and that's where that mRNA is going to pass through. This green area, which is sort of the active site. And then if you notice, there's also, it looks like there's DNA in here, and it's not DNA, it's actually RNA. And you'll read in your book about something called rRNA, or ribosomal RNA. And that's what this is here. So this ribosome, which takes RNA and turns it into protein, is actually a complex of RNA and proteins. And so I guess it's a good middle ground for us. But this is a really complex structure here. So I just want to give us a view like that. We could look at it like this as well. Here's just another artistic version of it. Again, you see the two subunits. You see the different colors to indicate the different strains. Here's the active site where the real action is going to take place. Okay, and then here's a really, really simple view of it that we'll see in a lot of our textbooks, and this is fine too for the purpose of it. We see that it's got a smaller and a bigger unit. We see that the mRNA moves through the ribosome, and then we see that as that RNA moves through, one section at a time, each section is going to contribute an amino acid that's ultimately going to build a bigger protein. So that's what ribosomes are like. They're responsible for protein synthesis, which means making proteins. It does so by reading the mRNA and matching that sequence of nucleotides to corresponding amino acids. So questions that you ought to have maybe, like how does a ribosome actually build the protein, and how does it know how to do that? So we've got to answer those questions as well. Of translation. So what happens at that ribosome, and then what's going to happen to that protein going forward. Okay, so I hope this video is helpful for you. Send me questions if you've got them, and I'll see you in class. Thanks.